that I don't kill your brain. I was gonna get into selfish DNA, but I think I'll give you like a week of break and then get back to it. So I think I'll start with a small update, and then from there we'll talk about two, let's say, big terms that are important for genomics, ENCODE and GWAS. So I talked about them a little bit 2 weeks ago, but I mean, I can't let you leave this class without talking about these two big things, okay? Before that, let's just see what's left. So the hardest part for the course is done. You've done so many assignments that now there's only 2 left plus the final, okay? The reason why there's a lot of assignments like this, I used to like it when professors split everything into smaller pieces. So if you destroy one of them, you can still, make it. So you'll have a second article assigned. This one is much simpler than the first one. Along with the corrections, you should be able to, I'm pretty sure you did well on the first one, but this one's probably going to be better. So it's again the same story, 5 pages, 4 to 5 pages. Just read the article and re-explain it. It's more in the form of a review paper than an article. Although it is a research paper, it's much more simpler, okay? There's no CRISPR-I and DepMap and whatnot, okay? So that's that. You have until November 15th to submit it. One important thing for this assignment, because it is on the Neanderthal topic, I want it to be submitted before the lecture, okay? Not at midnight. So I'll send a reminder about this. Next lecture, we'll talk about the 2 things that we didn't, one of the 2 things that we left. So we talked about genes, regulatory regions, and all that. Now we will focus more on selfish DNA and functional RNAs. And this is just a short survey of, because they're much more complex than genes, I could go on just for a full course just on these 2 topics, okay? So I'll do a survey on like microRNAs and shRNAs and things like this. Today we'll talk about 2 technologies, well, one consortium and one way of thinking about data that is very important for genomics and that you can pretty much use right away. And then we'll get into the kind of interesting topics. So cancer, you already read the paper. Neanderthal, you'll read the paper. I'll be talking about them. And then I'll be talking also about gene therapy. As you've heard me throughout the semester, I've been talking about cystic fibrosis. So I think I'll use that example for this lecture. And then the microbiome, so with a view on the genomics of the microbiome, and you'll see how interesting it is and what you can learn from there. Then it is the presentations by the students. So I forgot what I said. Did I say 20 minutes? I think I said 20 minutes per presentation, right? Because there's, no, 30 minutes. 30 minutes per presentation, so we'll have 2 groups each lecture. That should fit the whole class. So in the presentations, we'll be drawing basically who's presenting first. If you want to go first, you can tell me. If you don't want to go first, then we'll pick you up at some point. Okay? What else? Final exam. Final exam focuses more on this part here than on the previous part. The presentations are not included in the exam. The papers are included, but what I'm going to be talking about, right? So you'll read them, you'll write about them, and then I'll talk about them, so you should be pretty good for these questions. And that should be it, I guess. And then the final exam. Final exam is the same format, and it's not much longer. I think there's like 2 questions more or something like this, okay? Just a little bit more space for writing, and that's it. So, questions for me? No? Okay. Oh yeah, please try to not miss the presentations. So, because if you miss them, and let's say you have a medical note, then if you want your grade for that, we'll present it one-on-one, and you'll present the whole paper by yourself. So try to not be sick. If you're still sick, then obviously we can't do anything. We have to do it that way. The other team members, if let's say, for example, it's a team of 3 and we're missing 1 person because that person decided to not work, because that person decided to be sick, because that person is actually sick, then the other team has to present the other members have to present that part, okay? please try to avoid that. Obviously, if you're sick, then, you know, this is life, right? What else? This is the paper that you'll be reading. So it's about the Neanderthal genome. So you've seen this in humans, you've seen this for yeast, and now I want you to do some work related to Neanderthal from what you learned and obviously from what they're talking about in the paper, okay? So the concept of gene keeps changing, and we still don't have a perfect explanation for it. Although you might think that we do and we understand it and we've been studying it this way, it keeps changing because we keep discovering things about genes that, about sequences that actually fit within the description of a gene, or we keep updating it, okay? There is one project which is run by a consortium called the ENCODE project, which basically took the sequence coming out from the Human Genome Project and the Celera Genomics sequencing and tried to associate function to almost each nucleotide, okay? And so these guys here, they had a lot of controversy because you'll see in the lecture I talk about it. Basically, their definition of genes meant that we had to rewrite all the textbooks. And so the community was like, nope, not going to happen. So they started making fun of them. And you'll see one article that I'm presenting how the scientists can be quite sassy. Actually, this is scientists have a way of, of telling you you're not doing it properly. And then I also want to talk quickly about genome-wide association studies. This is more like a method of working, so we use it in research. This is a consortium, so they have a website and everything. GWAS is a way of working that allows you to study complex function of DNA in a pathological state, okay? So ENCODE, let's, you know what, let me open the internet first. The browser, I mean, still 1993 saying open the internet. ENCODEproject.org, I think it's .org. Right, so this is a team. Well, it's not a small team, it's a huge team. There's many universities that are involved. There's many professors working on this, many labs, and they have different data centers. And what they do is that they will basically try to understand how the DNA is structured and what are the parts of the genome that has been sequenced by the HGP, right? And so they try to assign a function, whatever it is, to each part of the DNA. So for that, what they do is that they take the DNA that was sequenced and then they run it through multiple tests to test for different things. For example, if you do an RNA-seq, you'll see what genes are being expressed because this looks at the set of transcripts in the cell at a specific moment under specific conditions. And so that will tell you what parts of the genome in that setting do. If you do ChIP-seq, for example, you're basically looking at binding of transcription factors at different places in the DNA. And so this will under, again, specific settings, specific conditions, and that will tell you where there are protein binding domains on the DNA, right? And then you have a lot of different other tests. For example, how open is the chromatin? You can do that with ChIP-seq. Also, you could look at coiling. You could look at what else? The different regulatory elements. For example, the enhancers and how they're linked to different genes and things like this. So these guys, data is still coming in, and so they're using it to determine what the genome does and what is the function of the sequence that has been brought up by the Human Genome Project. So the initial lessons from ENCODE, that was in like 2007, I think, or something like this, right after the sequence of the human genome was available, they discovered that actually genes are not discrete units. What do I mean by discrete units? So if you imagine the DNA sequence like this, from here to here, it could be 2 genes, 3 genes, and they could be overlapping. So it's not like one gene, one regulatory element, one gene, one regulatory element. Instead, you can have a gene overlapped with the other one, and depending on which promoter is being used, then you'll end up coding for protein A or protein B, okay? For example, in this segment here, there are 4 genes. And so remember, DNA is double strand. So you have a forward and a reverse sequence. So the forward reverse could have a specific set of genes, right? Because the ATG would be read this way. But if you look at the reverse sequence, you have a different set of genes because you're reading it from the other side. And sometimes you'll create these ATGs, which will lead to a protein, okay? For example, in this case here, in this fragment here on chromosome 21, you have 4 genes that are being that are placed here. The first 2 are on the positive strand of DNA, so it's ITSN and crazy L1. And then on the negative strand, you have Donson and ATP50. So you can see how the same segment of DNA has different genes based on the strand that is being transcribed. That's the first thing. Second thing, they also figured out that genes overlap. So you can see how Donson here and ATP50 are super far apart between each other. So there's about 300 bases between them. But it seems like these 2 genes create a transcript that contains both genes at the same time in it. So you have exons from one and exons from the other one, okay? So this is bringing a level of complexity that requires a lot of computational power to determine. And this is one of the role of ENCODE. Okay. They also discovered that there are these regions called transcriptional activation regions. There's a mistake here. Transcriptional activation regions, which were not identified previously, but they have a functional role in the DNA. For example, you have here these regions here that are between genes that don't seem to have any function, but as you do all these analyses, you realize that proteins actually bind there. And so, for example, this could be an enhancer, right? And this enhancer here is between gene 1 and 2, so it could actually be regulating both genes at the same time, okay? So this is a functional role for this gene. And then obviously, if you look at the opposite strand, you have a different set of genes. Although they're complementary, they don't code for the same genes, okay? Now, there is a model for these transcriptional activation regions, which were annotated by ENCODE. And what they're saying here is that, let's say, for example, on this strand, you have 3 genes, and you have these transcriptional activation regions between them. Then when you're making the transcripts, the way we used to see it is that you would make a transcript only for gene 1, only for gene 2, and only for gene 4, right? But in reality, you can make also a transcript that codes for 2 proteins at the same time. So this is the new things that were kind of discovered by the ENCODE project. And so because this enhancer regulates both of these genes, then usually it gets transcribed together. And it gets transcribed together, producing kind of these new transcripts that have exons from gene 1 and exons from gene 2. Why does this happen? There are some genes that require expression at the same time. And so the way you express them at the same time is by controlling them at the same time. And the way you control them at the same time is by having one enhancer regulate both genes and have them both transcribed together in one unit, right? So you can take a look at this picture, and you can see all the different combinations that come out from this. And obviously, don't forget alternative splicing. For example, here there's only exon 1, then this enhancer here, and then let's just call them transcriptional activation regions because they can be also insulators, silencers, repressors, whatever. Okay. What else? What else? Yep.
So are these, like if you take one of the green exons and 4 red exons,
yep.
Is that making one protein or are those
2
separate proteins?
2 separate proteins. So this one here is only has exon 1 and produces a functional protein. And this one here has exon 1, 2, 3, and 4. Right? And then you have multiple different combinations depending on the state that the cell is in. Okay, so I've been talking about a gene, but we still don't know what it is. So there's this Gerstein guy that proposed something which is still valid. And he says that a gene is a genomic sequence, either DNA or RNA, that can encode a functional product. So that means either RNA or protein. So this is kind of basic, makes sense. We're used to the protein explanation, one gene, one protein. But then also RNA is important. So you have genes that don't produce proteins, but they do produce RNA, non-coding RNA. And this RNA is important. We'll see it in the next 3 lectures. or 2 lectures. If you have a case where you have multiple functional products that are shared, that share overlapping region, then you consider them as one gene, okay? So, for example, in this case here, okay? Even though they're making multiple transcripts, they're still part of the same gene, okay? And then last, let's say postulate, or whatever you want to call it, you can't combine RNA and proteins. So when we're talking about a gene, that gene produces either a protein or an RNA, and you have to be coherent. Even though they share the same sequence, the RNA gene, the gene that codes for the non-coding RNA can be this long, and then part of it codes for a protein, while those two are 2 genes. Okay, so these are the basic rules. You can read the paper here. Nothing too interesting, but this is basically the summary from that paper. There's another example from that paper where you have a DNA molecule, a double strand that has 5 genes. So these A, B, C, D, and E. And so when you make the primary transcripts, for example, you can be producing a transcript that has A, B, and C together because they are regulated by the same enhancer, let's say, or same multiple promoters but same enhancer. And you can have another transcript which has the other 2 genes, but then this whole segment as well can code for non-coding RNA. So there are multiple genes here, not only 5. There is 7 products that come out from here. Okay, 7 different. No, not 7 different products. Sorry. So you have 1, 2, 3, and 4. This is just a random example. Okay, it's just showing you that after you splice these segments here from the same sequence here, you can produce a non-coding RNA molecule, and you could also produce proteins depending on what is transcribed. Okay, and obviously what is transcribed depends on the state of the cell. If the cell needs a non-coding RNA, then it will make the non-coding RNA from this sequence. If it needs this kind of protein that has these 2 genes, this transcript that has these 2 genes together, then it will make this one, etc. Okay. Usually what happens is that the non-coding RNA is used to silence the coding RNA. So depending on what gets activated, because they have the same sequence, right? So depending on whatever is being activated, if you have too much of that protein, then you'll make the non-coding RNA that will destroy the transcripts that lead to making protein. Okay. Obviously, it's much more complex than this. So we used to see this as one sequence, one transcript, one protein. But in reality, it's not like that. Sometimes you have one transcript, 3 proteins, because they need to be expressed at the same time. Sometimes you have one transcript, no protein. And so the central dogma still exists, but it has changed. Okay, it's not 1, 1, 1. The ratios have changed. Okay, so that was my little intro about what is a gene. This is the current state of what is a gene. There's a lot more being studied. Obviously, we have so much DNA that we don't know what it does, that we're still trying to understand. Okay. So a little bit about the 2 projects. So we said that we have the sequence, and we're trying now to identify its structure. So we looked at introns, exons, promoters. This is all the stuff that I talked about before, and I added a little bit to this today. But for the functional annotation, I went kind of quickly over it. So if you imagine each piece of DNA has a function, even a single nucleotide has a function, okay? Then you need a way to kind of associate a function to the piece of DNA that you're trying to study. And there are many tools that exist, thousands, if not tens of thousands of tools that have been developed to do this. They require some sort of data that is produced in the lab and then used to infer function. But as time goes, computational methods have now appeared, which kind of take the sequence and compare it to other sequences and then try to infer from there. So the way you assign function to genes is actually quite simple, but sometimes not so obvious. So depending on what gene you were working with or what piece of DNA you were working with, you can kind of look at other genes that look like it and try to understand what it does, right? For example, if you have a protein that is important for iron metabolism, there isn't only one protein that is important for iron metabolism. There's many proteins that are important for iron metabolism, and some of them, they do have a site that binds iron. And so if you find that site in other proteins, probably it's part of iron metabolism. Obviously, this is not super accurate, but it's an initial starting point from where you can start inferring function. If you're studying iron metabolism and you're looking for proteins that are important for iron metabolism, you need to find that kind of structure. If you don't, probably it's involved in another process, right? And as I mentioned previously, we have domains in proteins and motifs that are important. For example, the DNA binding domain. If you find, let's say, for example, find a group of hormone-dependent nuclear receptors, they detect hormones and they act as transcription factors, then you'll probably find a hormone binding domain. And if they bind to DNA, then you'll probably find a DNA binding domain. And that DNA binding domain potentially has a motif, an amino acid sequence that binds to a specific DNA sequence. So these are known as, for example, the zinc fingers or whatever, okay? So this is an initial kind of dirty approach to find the function. So this is how you start annotating the genome. But obviously from there you need a little bit more specificity. And the specificity comes from experimentation. You cannot confirm a protein function just from computational methods. Usually the way it goes through is that you have a sequence, compare it to everything that you know, BLAST, align, whatever. You find the domains, then you say predicted function. So in the database, it will be entered as predicted function. As you do experiments to it, then you'll figure out that actually it is that function or not. Then you can turn it into curated function, okay? So there are these databases that act as gene ontologies, and they tell you what the function of a gene is. And this is important because some people in science are a little bit funny. So they create these weird names, and just from the name, you will not find out what something is. So you need these gene ontologies to tell you what the functions are, where the protein localizes, and things like this. One of the groups that really likes to name their proteins in a weird way is Drosophila people, okay? If you look at the names of Drosophila genes, they're pretty crazy. I don't know what's wrong with them, but like if I give you the gene called son of 7-less, you have no clue what the hell that is, right? Right? But this person was probably like thinking about SOS and he couldn't find another, you know, and they have some very weird names, okay? There's a gene that I would love to show you, and it's actually very, how do I say that? Very stereotyped, limit insulting to people with disabilities, and it has passed in the kind of, what do you call it? In the literature, and it's being used, and it's like mother of, and just, you know, crazy, okay? Huh? Yeah, exactly. Just don't. Yeah, this is a really bad name, okay? A really bad name for the gene. Like, it's horrible. I don't know how it's accepted, but anyways, you can Google it later on. So, son of 7-less, its actual function is just a guanine nucleotide exchange factor. And this protein here, all it does is that it converts GTP to GDP and transfers the phosphate to a GTPase, okay? Now, if you want to go from one organism to the other, one species to the other, you also need these gene ontologies because some different groups name their genes in different ways. For example, in yeast, RAS GTPase is a human oncogene, but for yeast it's called CDC25. CDC25 comes from cell division cycle protein number 25, and it's just the mutant 25 that blocked the division, right? And so they called it CDC25. Yeast people are quite boring. My PhD was on yeast. Seems like Drosophila have it better. I don't know why. Yeah, so this is why these gene ontologies exist. So There are three categories that are being used for gene ontologies. So you have the molecular function, for example, phosphatase. You have the biological process, glycolysis, and you have the cellular component, nucleus, for example, okay? If we take a look at an example here, so this is on NCBI. If you go on their gene section and you look for SOX1, sex-determining region Y box 1 gene, you can take a look at the GO for its molecular function, and you can see that it's DNA binding, so you know that this localizes on DNA, and you can go more specifically into the functions that have been associated to it. There's always an evidence code. For example, these evidence codes are used everywhere, so maybe it's important that you remember some of them. For example, this is a direct assay, so you know this is solid. They tested it in the lab. This is a binding assay, things like this, okay? You also have the process that it is involved in. So here you'll see there's a lot of variety. The reason for that is a protein is involved in many processes. For example, brain development, binding assay, direct assay, mutant phenotype. I forgot what the I is, but basically figured out through the mutant phenotype, okay? And you can take a look at all that. And then if we have DNA binding, well, obviously, you'll probably find it in the nucleus, okay? So when do you use Go terms? So instead of going through this slide, I will actually show you an example that you can understand a little bit how interesting this is. But for example, if we're studying something and we're treating the cells with a specific molecule, and we want to understand how that molecule ends up killing the cell, for example, in the case of cancer, then you'll add the drug, you'll take all the transcripts that were expressed in the control compared to the experimental, which is your drug, let's say compound 75, which you're studying right now. Then you'll extract the transcripts, sequence them, and then you'll find that some genes have been upregulated. Their expression has been upregulated. So then, if you have only 2 genes, very simple to understand, right? But if you have thousands of genes, you need some sort of computational way to do this to tell you which Go terms are being enriched. And you can use these Go terms to then direct your research. If you see that 80% of your genes have been enriched, are in iron metabolism, don't go and study glycolysis, you know. It's probably through iron metabolism that the cell death is happening, okay? So I'll show you an example here. There are many tools, but this is just one called Enricher. So me, when I do, for example, mass spec experiments or RNA-seq, ChIP-seq, whatever, I will see what's being enriched, let's say, for example, twofold. Then I will take the group that is being enriched, and I will put all the genes in here. And then I will submit this, and it will tell me, let's take an example for breast cancer. Nothing to analyze. What do you mean, bro? Wait. First time I try their examples. Oh, okay. So the genes appear like this. I had to wait. Jeez. So the genes that have been enriched in breast cancer, let's say, experiment are these. Then if I submit it, it will go here and give me different databases and their different Go terms that they use. For instance, if we look at disease and drugs, let's see, hopefully breast cancer comes up. So breast cancer comes up, neoplasm comes up. So you can see things like this. If you want to look at the pathways that have been upregulated, so then you can target them with a drug, you would come to pathways, and then you'll see that for this specific set, based on these conditions, this person has an increase in the interleukin signaling pathway, right? You can also see, let's see, apoptosis. So now you see apoptosis, for example, here. So it doesn't mean that apoptosis has been upregulated. It means that maybe proteins that inhibit apoptosis have been upregulated. So you can think breast cancer, unlimited cell division, apoptosis appears here, so probably there's an increase in the inhibition proteins for apoptosis, okay? So this is what I mean by the Go terms, and this is a good starting point for studying anything. So if you're doing these genomic studies, you'll be using this a lot. If you want to find the transcription factor that is involved at the top that controls all this, Then you go to transcription, and you see that this looks like an AR chip experiment. So maybe in this breast cancer, androgen receptor is causing the problem, right? And so now you can try androgen deprivation therapy or treat with androgen, things like this, okay? So that's that. Obviously, you see P53, you know it, right? You've studied it many times. And things of the sort. If you are doing bioengineering and you want to upregulate a specific pathway, then you can do this. You do an RNA-seq experiment and you see if your lipid metabolism has increased or not. This can do that for you, and you know if it's working or not. Okay? Good. So ENCODE. ENCODE started, well, 2007, but 2009, the data, I guess they started accumulating data. So through the years, there's a lot. They started integrating multiple different sets of data coming from different studies. This is very hard to do. So you're basically trying to understand how DNA is structured and its function by incorporating, for example, histone ChIP-seqs, which tell you if the chromatin is open or not. So if it's a transcriptionally active region, transcription factor ChIP-seq, which tells you where the proteins bind, your transcription factors, RNA, where was it? I guess they put RNA-seq in this one. Knockdown RNA-seq. So this is telling you which transcripts that are being upregulated or downregulated, and then you can figure out the function of each piece through this. Obviously, they have only about 9,000 assays that have been done in human and mouse, but this is growing super fast. Because of all this data, what happened is that they went in a frenzy kind of and associated functions to everything, okay? And so they used this in a bad faith way, and they started publishing papers nonstop. And that's because they were saying, not thinking, that 80% of the human genome has at least one biochemical pathway that it works in, and that has some sort of chromatin-associated event in at least one cell type. This is not true, okay? No way that 80% of the DNA has a function, well, at least from what we know now, right? And they said that 95% of the genome lies within 8 kilobases from DNA-protein interactions, so they think that because there's a protein interacting with it, that probably there is a function, okay? And that 99% of it is within 1.7 kb of a biochemical event, so it must have a certain function, right? Biochemical event is, for example, methylation or acetylation or whatever to open and close the DNA. And so this led to a little bit of controversy, because that means that basically we had the junk DNA theory wrong. We don't understand what genes are, things like this. And they were kind of generous in the way that they were saying that the DNA has each piece has a functional role to it, because they had two rules. The first one makes sense, which is defined product. So each piece, if it has a defined product, it must be gene, so it must have a function, being a protein or non-coding RNA. It's the second one that is a little bit more generous, where they say that basically there is a reproducible biochemical signature, for example, protein binding, so it must have a function. But protein binding does not mean that you have a function. It's not always the case. And if a structure of the chromatin is open or not, it does not mean that there is a function associated to that, right? I'll give you, I know this is a little bit not clear, but I'll give you examples so you understand. There are hundreds of questions that were asked. There was a big fight around this. But if you have many of the events that were talked about by ENCODE are transcriptional, not translational. And so how can you associate a function to a transposon if its role is just to be selfish? Right? It has no human function, a transposon, but they associate to it a function, and then they publish a paper on it. But that's not really human-specific function, right? And then they also did some evolutionary studies. So these evolutionary biologists that did not like this idea, they were against it, and they said that how come they're not under selective constraints, these pieces that you're assigning function to? If they have an important function, they must be selected for or against. But when they did DNA analysis of different organisms, they see that there is no selective constraints. So is there a function? Is there not a function? So this led to this newspaper, I think, or it was on TV. But basically, the evolutionary biologist went on and said, we conclude that the ENCODE consortium has so far failed to provide a compelling reason to abandon the way we think about function in the genome. And then, obviously, they're salty, so they wrote or said, we agree that many, because the people, these ENCODE people were basically saying everything that you wrote in the textbooks is not good, so you have to change all the textbooks. Obviously, no one is going to do that. So they said, the evolutionary biologist said, we agree that many textbooks dealing with marketing, because they're talking just about the hype around the ENCODE, media hype and public relations may well have to be rewritten. But they're really talking about these guys here, okay? A little bit salty, but it's okay. So ENCODE then went on and decided to fix their little bit of, let's say, inflating the data to review a few things that were challenged by the evolutionary biologist. Transcription does not equal function, not all the time. Sometimes you don't produce anything functional. Histone modification, so opening or closing of chromatin does not mean that there is a function associated to it, and things of the sort. Now here I just put all the things that were criticized. It's just for, the purpose of telling you what the hell was happening. But obviously the ENCODE critics and all that, they're not important for you. Okay, it's just to give a little perspective in history of what happened. There's a cooler group or a cooler way of doing it. So the ENCODE still exists, and now it's a little bit more legit than it used to be. They got a lot of Nature papers from their work. Some of them have been retracted, things like this. So there is another approach that was used for doing these studies to associate function to different pieces of DNA, and this is called genome-wide association studies. So this is not a consortium like ENCODE, but instead it is just a way of working, okay? And in this case, what you do is you take information from two sets of individuals, one that is healthy or has the trait that you want, and the other that does not have it. They have to be not stratified. That means you don't take the same group and then take half of it, give them the trait, and the others don't. Instead, you just take them from the population, and then you try to find what genes or what pieces of DNA give that trait, right, by sequencing both. And this usually occurs through single nucleotide polymorphisms. And you can pretty much study anything, okay? So the way GWAS works is that you'll collect data from people, you'll genotype, so genotyping just means figuring out their DNA sequence. And then because you have data from other groups, then you can do these clustering analysis, which will tell you, for example, if you're, in this case, they're studying, I don't know, probably like ancestry or things like this. So it will tell you based on your sequence where you are. Are you more closer to African, Asian, American, whatever? But in reality, for example, you'll have two groups. You'll have the healthy disease states, and then your data will be clustered closer to the one that you represent. From there, then you can actually look at the DNA specifically on a single base approach and see where actually this trait comes from. So you'll see that some nucleotides, you'll see them different between the two groups, and because they are different, then you will understand that probably that's where the trait comes from, that single nucleotide polymorphism, okay? And then you can do different analysis, and then just repeat and keep doing that, okay? So the goal of GWAS is to go after genetically linked disease, and there's a lot of them that don't follow your Mendelian inheritance, so 3 to 1, 1 to 2 to 1, whatever inheritance. One example is, for example, height. Height is not a Mendelian inherited. It's not inherited in a Mendelian way. Instead, there is, I think, like 1,200 SNPs that are involved in height. And it's a highly, they call it a highly heritable trait, obviously. But some of them are less heritable. Okay, so there is a depth that can happen in terms of traits. height, you kind of have no choice. So there's more involved to it than other traits, okay? Some diseases that are genetically linked, usually heart disease, macular degeneration, Crohn's disease, celiac disease, which I have, keratoconus, which I have. So if you don't know what keratoconus is, basically your cornea becomes a cone, and you have to do surgeries to fix it all the time, but you lose sight over time and then repeat. There's a lot more: cystic fibrosis, etc., cancer, you can name them. There's a lot of inherited diseases that come from inbreeding, okay? This is a weird topic, but still happens in the world, you know, in family, or inside the family. So here in Canada, for example, it's illegal, and you have to measure the degree, how far you are from, let's say, ex-cousin or second-generation cousin to be able to do that. Otherwise, you could take, I think, like 10 years of jail or something like this, just for this, okay? But there are places in the world where this still happens, and this leads to an increase in terms of these genetically inherited diseases, okay? What else did I want to say about this? I forgot, but I think that's enough information for you. What else? So SNPs, so when you're doing these GWAS, you're not going to get everything that is related to a trait because you don't have 8 billion people in your study. Instead, you'll have a portion of it. So you try to find the SNP that has the most effect on the trait, but obviously the more your sample is bigger, the bigger your sample, the bigger the amount of SNPs linked to that trait. So this is what it looks like. You will sequence all the chromosomes and put them in a line like this. Compare the two states and see where the differences occur. Let's say this is disease X, okay? So you see here, for example, that there is a high amount of variation at this loci here. And so you can take a look at this one here and see what, let's say, what nucleotide is changing. Now, in this picture here, I don't know why they took chromosome 15, but let's assume this one is the most varied part of the sequence. Then you can take a look at it and you'll see the chromosomal position where there is a lot of change between the different organisms, the different individuals. So when you find the difference between them, then you can study this more in relationship to the genes, to how open is your chromatin, to things like this, right? So if you find your SNP here, what gene is it close to? Is it in a region where there is an enhancer? Is it a region where a transcription factor binds? And if so, how does that binding change based on this polymorphism, right? And then, obviously, you can link it to a specific gene then by just doing some mutations to it, removing this part of the DNA and things like this, right? For height, we thought that it was controlled by a very low number of loci, but as you add in the sample size, it becomes kind of exponential, the number of SNPs linked to it. So as you can see here, after adding up to, let's say, 800, there's now about, 2,200, 2,300 SNPs that are linked to height, okay? So maybe one adds 1 millimeter, the other 5, things like this, okay? So that's that. This is the last slide, then I will go over the exam quickly, and then we can go home, okay? Recent list of successes from GWAS studies. So I use GWAS in my research, but everyone is basically using it, okay? You don't have time to go and look for a specific nucleotide, just take the whole thing, sequence it, compare, and let the computer do it, okay? One that is very interesting that I want to focus on is educational attainment, okay? So educational attainment means how does your DNA affect how far you get into education, okay? This is kind of controversial. I talked about something like this in my other class, and I got a comment from a student. I guess the student did not, or felt like I was saying something, but I was not saying something. So I will repeat it in a different way, okay? Because I don't care. But you remember Watson and Crick? You know what happened with Watson? Okay, so Watson got the Nobel Prize for the DNA structure, and then he lost it. The reason why he lost it is because he was saying that race and skin color affects intelligence, right? And so obviously people were like, nope, not happening. So there is research that happened on that. They didn't find anything that is kind of saying the exact same thing as him. Science community was like, nope, no Nobel Prize, no any prize, that's it. And now he's basically kind of useless. But which brings me to this point here, educational attainment. This is kind of saying the same thing, right? So sometimes you will see research like this published in Nature. So if you click on 23, you can read it. This is published on Nature, okay? And it's basically saying that there are different SNPs that affect your educational attainment. Obviously, you guys see why this is stupid, right? Because if you're born inside of Harvard or in Batnamay City, you're not in the same opportunities, right? So educational attainment is dependent on many other things other than just your SNPs. So I don't know how this paper got published. This is pure bullshit. Don't agree with this stuff. You can read it to see how sometimes science is a bit weird.
Could it be a part of the factors, just not the whole thing?
Of course, because the environment kind of affects your genes, right? Epigenetics, right? And so, of course, that if after 200,000 years, you're only going to Harvard, and Harvard is a top school, and the other one is living in a poor country with like no water, obviously educational attainment is going to change your DNA, right? But it's not a major part. It's a very small part, but you can do these kinds of studies. As a matter of fact, you can do them at home because you can take sets from different groups, do research, and it's all in the same database. And then let's say, for example, you want to compare whatever things. You just filter out, you take your two data sets, compare them, and it will tell you where the changes happen, right? So this is a good starting point for anybody's research that wants to do this kind of work. If you have some crazy ideas that you want to test without telling people about, you can kind of do that. Yeah, so that's my point on GWAS. Next lecture, we'll be talking about selfish DNA. And then 2 more lectures on functional, and then that's it. The boring topics, we'll get into the more fun stuff, okay? Questions for me? Please don't leave a comment saying that I'm racist or whatever. I'm Algerian, okay? I'm getting those problems myself, you know.